What is cladistics? A burning question in all of your minds, I know, and something that you've all kind of wanted me to explain, also known as phylogenetics and systematics. Cladistics is the study of the evolutionary relationships between organisms, an organism being a living thing. And it maps this using phylogenetic trees, aka cladograms, basically diagrams of how different organisms are related like a family tree. There's a lot of good evidence that all life on Earth today evolved from a single common ancestor, and that single common ancestor had a cell membrane, genetic material, liquid inside of the cell membrane, and proteins to do things like copy that genetic material and metabolize energy. So scientists have one burning question based on this information: how did life evolve? Now, scientists figure out how life evolved using a variety of different types of data, such as genes, fossils, and a whole bunch more like proteins and metabolomes and a whole bunch of other stuff that we're not going to get into. The big ones are genetics and fossils. And we use these different types of data to figure out how things evolved and then to classify them based on that evolution. Now, in the olden days, back before we knew about evolution, we classified organisms based on traits. Certain traits signified new types of organisms. Certain organisms were more high up than others, and there were different ranks of organisms based on inclusivity. And this was dubbed Linnaean taxonomy, which is that system that you learned in school. Domain, kingdom, phylum, class, order, family, genus, species. Yeah, no. Why not? Well, it's only based on living life, so it doesn't actually take into account transitional forms. It doesn't accurately reflect evolution at all. It's completely arbitrary and based on traits that we designated as important or not, and lots of organisms are the only members of their family or even order because everyone must have a family or order. But more than any of that, it's not scientifically testable. It's based on arbitrary human judgment, so for the question, is Archaeopteryx a bird or a reptile, the answer doesn't actually matter because it's based on what you think makes a bird a bird. So someone could be like, Archaeopteryx has feathers, that means it's a bird, and someone else can be like, well, it has a bony tail, it can't fly, and it has teeth, and be like, it's not a bird, it's a reptile, and then poor Archaeopteryx is given an identity crisis because there is no correct answer to that question. But evolutionary relationships can be tested. Is organism A more closely related to organism B than to organism C? We can actually test that using the scientific method and looking at how groups of organisms evolved over time. We do this by tracking traits, yes, but I swear it's not the same thing as Linnaeus. Rather than just arbitrarily giving some traits more importance than others, it's more of a sum of as many traits as you can possibly examine to look at the simplest explanation for a set of traits in given organisms. For example, both dinosaurs and humans are bipedal, but humans and chimps have way more traits in common than humans and dinosaurs do. It's really about what explanation is most likely. In this case, humans and chimps have more in common, so it's much more likely that they are more closely related than humans and dinosaurs are. Many traits evolved multiple times, in fact, because they're useful. This idea is called convergence, but sharing a lot of traits usually means a common ancestor. It's hard to have that many coincidences in terms of convergent evolution. Now, the bulk of the rest of this video is going to be me defining cladistic terms because this is the science of defining life, so a lot of it is just definitions, and I'm sorry, there is no way around it. I'm going to try to make it as exciting as possible. Whee! Here we go. A clade is a single unit in cladistics, and it's the most important unit. It's also called monophyletic, and it's basically a common ancestor for a group of organisms and all of that common ancestor's descendants. A really common example of this is Dinosauria. Dinosauria is just all the descendants of a single common ancestor as shown by this very simple circle. In my handy cladogram here, a clade is anything that includes an ancestor and all that ancestor's descendants. So this circle is the clade of I and all of I's descendants. This one is the clade of A and B. This one is the clade of F, G, H. And this one is the clade of E, F, G, H. All of these are clades because they include a common ancestor and all of that common ancestor's descendants. It's really not that hard to define a clade. Now, a paraphyletic group isn't a clade. It's almost all of the descendants of a single common ancestor, but it typically leaves some out because they've either lost or gained some trait that the ancestral organism for that group that they're a part of had. It's useful to talk about these because it's kind of like an evolutionary grade. For example, non-land-dwelling chordates, fish, include all members of chordata that are not tetrapods, which isn't a clade, but it's typically useful to talk about because it helps us to examine the ancestral traits for chordata. So it's very easy to define a paraphyletic group on a regular cladogram. Just circle some, but not all, the descendants of a common ancestor. Now, a polyphyletic group is anything that isn't monophyletic or paraphyletic, and it typically includes a bunch of organisms that do not descend from a very recent common ancestor. Typically, they share some sort of trait that evolved multiple times, And it's the kind of thing that you find in Linnaean classification because he'll group things together for sharing traits even though they don't have a recent common ancestor. For example, all flying chordates include abelae, which are birds for the most part, pterosaurs, and then a specific group of mammals called bats are all flying members of chordata, but they do not share a recent common ancestor, so this is a polyphyletic group. But wait, you say, isn't all life descended from a common ancestor? Wouldn't that make all polyphyletic groups necessarily paraphyletic? Shut up! I get where you're coming from, but sometimes it's more convenient to call a group polyphyletic than paraphyletic, and the reverse, depending on the context you're looking at the groups in. So for example, here, A and F are paraphyletic, but they're also polyphyletic. Let's move on. A character is a trait that is possessed by an organism that we use to define phylogenetic trees. A good example of a trait is having your egg in an amniotic sac. Now, a character state is the way that we describe a certain character. So, for example, we can describe it whether they're present or absent, or we can describe natural variations in a population. So for the first example, whether or not you have an amniotic sac, and for the second, the length of quills. See, it's really important that characters reflect biological realities. They need to be easily compared between different groups. They need to take into account the idea of epigenetics, for example. Whether a trait is present and its different forms are two different things. For example, does a mouse's fur have color? Yes or no? And is that color brown or gray? You need to have color to have that second trait, but you shouldn't lump them both together. You examine them as two separate characters because that's how genes work. They work in networks. Now, a taxon is a named group of organisms. It might not actually be a clade, but a clade is an actual evolutionary thing. Warning, the following terms are relative. Depends on the context you're using them in. A plesiomorphy in one context might be a synapomorphy in another. Let's move on. A plesiomorphy is an ancestral character, one that is retained by a taxon from its ancestors. Not all members of a clade might still have this character, but it was present in the common ancestor. For example, an amniotic sac is the plesiomorphy for all members of Amniota. Now, a simple plesiomorphy is a kind of plesiomorphy, which is an ancestral trait that is retained by the taxon you're looking at. It is not necessarily retained by all members of a clade. For example, the ancestral sauropsids were cold-blooded, but birds, which are a part of Sauropsida, are warm-blooded. So cold-bloodedness is a simple plesiomorphy of traditional reptiles. Now, an apomorphy is a derived character, a new state, or an innovation. It's used to define a clade. For example, having mammary glands is an apomorphy of Mammalia compared to the group it's part of, Synapsida. Now, an autoapomorphy is a kind of apomorphy that is a character derived in a single group that you're looking at. It typically doesn't say anything about evolutionary relationships. For example, the brain-to-body ratio of Homo sapiens is an autoapomorphy of Homo sapiens. Here it is represented, it's only present in one group. Now, synapomorphy is a kind of apomorphy that defines a clade. Unlike autoapomorphies, it typically applies to a clade that is made up of more than one branch on a phylogenetic tree. For example, the notochord is a synapomorphy of chordata. As shown here, a synapomorphy would apply to multiple branches on a tree. Now, two traits are homologous if they are inherited from a common ancestor or that had that character or trait, or if they come from the same origin in that common ancestor. Traits can change over time, so sometimes it's hard to determine homology. For example, all feathers are homologous, but they often look quite different. Now, a homoplasy is a character shared by two or more organisms but is absent from their common ancestor. Thus, a homoplasy is a convergent trait. It evolved multiple times. It does not define clades in any form. For example, warm-bloodedness is a homoplasy for modern birds and mammals. Phew. But wait, there's more. A basal member of a clade is one that is close to the root of that clade, or it is the earliest diverging group from a clade. This is yet another relative term. An Eoraptor, for example, is a basal Saurischian. As shown here, it is the earliest diverging member of Saurischia. Now, derived is a term that can be used to describe both taxa and characters. A derived character is present in some later groups of a clade, but not all. It is typically a modified version of a primitive character, and it evolved much later than the ancestor of that clade. For example, Saurischia have the ancestral character of a forward towards the head facing pubis. However, in birds, a derived group of Saurischians, the pubis points backwards, which is a derived trait. Now, a derived taxon is a member of a clade in question that evolved later after the origin of the group than other taxa in question. Birds, like the crow corvus, are derived Saurischians, as shown here. Now, incertus sedis is a fancy Latin term that means of uncertain placement. It applies to animals where we don't really know where they fall in a clade. They could be basal, they could be derived, it's not entirely sure. For example, Kakuru, Ornitholestes, and Yaberlandia are all members of Maniraptora incertus sedis. We don't exactly know where they fall in Maniraptora, we just know that they are Maniraptorans. Now, a reference taxon or a specifier is a taxon that is used as a reference in a phylogenetic definition. For example, Abele is described as all dinosaurs more closely related to Archaeopteryx than to Troodon. So Archaeopteryx here is a reference taxon. So is Troodon. Now, an internal specifier is a reference taxon that is in the group that it's referencing. For example, Archaeopteryx is an internal specifier for Abele. Conversely, an external specifier is a reference taxon for a clade that is not part of the clade that it's referencing. For example, Troodon is an external specifier for Avale. Let's talk more about clades. Now, an apomorphy-based clade is the least common type of clade, and it's defined based on an apomorphy. For example, it's the first ancestor of A to possess B that is inherited by A, and all descendants of that ancestor. Tetrapods, the first ancestor of mice from which mice inherit their 4 limbs, and all descendants of that ancestor. Some taxa, like snake, can lose that trait. Now, a node is a type of clade that is based on the most recent common ancestor of 2 or more reference taxa and all of that ancestor's descendants, basically represented as C plus D. The most famous example is Dinosauria, which is the clade of Megalosaurus and Iguanodon, their most recent common ancestor, and all of that animal's descendants. Shown here, Dinosauria, Megalosauria is in this group, Iguanodon is in this group, so Dinosauria is the group that includes both of them, which is this thing. Now, a stem or a branch-based clade is basically all organisms that are more closely related to one taxa than to another. It's typically represented as E greater than F, or some variation thereupon, and a common example comes from our Ornithischia definition, which is all dinosaurs more closely related to Iguanodon than to Megalosaurus. See here? Ornithischians have the group with Iguanodon, but none of the things that are more closely related to Megalosaurus. And with Dinosauria, we see an example of a node-stem triplet, which is the most useful kind of clade. It's a trio of clades with complementary definition. So 1 node and 2 stems that make up that node are example, Dinosauria, the node is Dinosauria, and the 2 stems are Ornithischia and Saurischia. So for a more visual example, here the node is represented by I and the 2 branches, and the 2 stems are this one and this one. Now, a nested group is a taxon that is within another taxon. For example, modern birds, Neornithes, are firmly nested in Dinosauria, as shown here. Corvus, a modern bird, the crow, is firmly nested in Saurischia, which, as we know, is part of Dinosauria. In a similar vein, a parent taxon is a group that another taxon is a part of. So, for example, Dinosauria is the parent taxon to Saurischia, and Saurischia is a parent taxon of Neornithes, as you can see here. Now, a sister taxon is the closest relatives of a given phylogenetic unit. They have the same parent taxon. For example, Saurischia is sister to Ornithischia. Now, in the previous cladogram, Velociraptor and Corvus are sister taxa, but that's only because I examined so few animals. See here? Corvus and Velociraptor, sisters. Now, complementary definitions are definitions for clades that all use the same reference taxa. Node-stem triplets, for example, all use complementary definitions. In our example, Dinosauria, Saurischia, and Ornithischia all use Megalosaurus and Iguanodon. Now, a reciprocal definition is where 2 clades use the same reference taxa but in reverse order, such as the 2 stems of a node-stem triplet. For example, Ornithischia is the reciprocal taxon of Saurischia. Now, an outgroup is a taxon that is hypothesized to be less closely related to the taxa that one is looking at in a cladistic analysis than any of those taxa are to each other. So in our example, Eoraptor is an outgroup to the rest of Saurischia, which is actually called Eusaurischia. Now, an in-group is basically just the reverse of that. It's the set of taxa that are more closely related to each other than they are to the outgroup. In this case, the in-group is Eusaurischia. Now, a crown group is a clade or a taxon whose immediate sister taxon is extinct. For example, Neornithes is crown birds. It's the group of all birds that are alive today and their most recent common ancestor. As you can see here, all things most closely related to birds are extinct. Now, the pan or total group is the crown group and all organisms that are more closely related to that group than any other extant organisms. So Avimetatarsalia can be called pan-aves. It includes all animals that are more closely related to birds than to crocodiles. Shown here, if we just circle all of that, that's Avimetatarsalia. So then everything that's left over is called the stem group, which is a paraphyletic group that includes all members of the pan group except for the crown group. So in this case, stem aves would be all non-Neornithian Avimetatarsalia. Yes, this means you can call them birds, but you don't have to. Birds is arbitrary. Birds is Linnaeus. Basically, all Neornithes are definitely birds. Anything with Avimetatarsalia could be a bird, and what you decide is a bird or not is up to you because it's arbitrary. Yay, Linnaeus! Now you understand the what are birds problem from a cladistic perspective. You're welcome. So in our lovely cladogram here, stem aves are all of these things, the extinct members of pan aves. Don't worry guys, we're almost done. I'm sorry, this is a lot of definitions, but this is the science of defining life, okay? It's going to have a lot of definitions. In fact, that's almost all it is. Definitions! Now, parsimony is a scientific principle that we use to figure all of this out. It's basically the idea that the simplest explanation is the best possible explanation as long as it fits all of our observable data. For one of our first examples in this video, it's more simple for humans and chimps to be closely related than for humans to be more closely related to dinosaurs. Also, for example, it's much more simple for birds to be dinosaurs than to have evolved from any other group, because no other group shares as many characters with birds as dinosaurs do. Ta-da! It really is that simple. Now I'm going to briefly define some of the terms that we use to study whether or not a cladogram is good, and the math that we use to evaluate cladograms. Even though you might not necessarily need to know this, it's good to know the kind of ways that we evaluate these things. So the consistency index is the measure of how well a character fits on a phylogenetic trait. If it's not completely compatible with a trait, it will be less than 1. It decreases with an increasing number of taxa, so there are problems with using this value. Still used sometimes to examine a character's relationship to an established trait. Basically, it's the minimum number of changes in a dataset divided by the actual number of changes needed for a cladogram. Yay! Math! Now, a retention index, it measures how well synapomorphies explain a proposed cladistic trees. It basically takes the maximum number of changes on a tree minus the number of changes on the tree, and then divides it by the maximum number of changes on the tree minus the minimum number of changes to the data set. Now, the rescaled consistency index basically multiplies the CI by the RI. This allows for a CI to be stretched and give it a scale of 0 to 1. Thus, the homoplasy index is 1 minus CI. The homoplasy excess ratio is basically the measurement of the amount of homoplasy on a tree relative to the maximum amount that could theoretically be present. So it's 1 minus the observed homoplasy excess divided by the maximum homoplasy excess. A value of 1 is no homoplasy, a value of 0 is the same amount as you'd find in a random data set, and a negative value is too much homoplasy! Now, there are a lot more terms in cladistics, however, unless you plan on going into this field, you don't need them. Trust me, we're not going to go into them. Now, I know what you're thinking. Meg, I still see Linnaean classification. That's because we haven't figured out the entire phylogenetic tree of life yet. Do you realize how diverse life is today? Never mind the fact that life today is probably less than 1% of all life that has ever existed on the planet. Of course we're not done yet! We use Linnaean groups as placeholders until we can come up with a better phylogenetic or cladistic groups, and sometimes Linnaeus got it right, especially with larger groups. With the smallest groups, sometimes it's more useful to use Linnaean genera and species than it would be to use the smallest possible clade. And as for species themselves, species is a tool used in biology to talk about evolution and biodiversity. There are many different species concepts and each has their place, but each is arbitrary, so more on that on a later date. So we use cladistics to show how life has evolved over time. It shows that evolution is a tree, not some sort of strange stepladder. All modern life is equally derived from life's common ancestors, so all modern life is equally evolved, no matter how much we do or do not resemble that first cell. Similarly, branch length can be used to show the time since a branch split from another. It doesn't always if that's not the purpose of the cladogram, and it would be kind of hard to scale it, but it can be very useful. As shown here in my friend's cladogram, you can show how long it has been since different groups diverged from each other. Link to this cladogram in the description. Cladistics also shows that evolved from means that it's part of that bigger group. Birds are dinosaurs because they evolved from dinosaurs. Neornithiaceae is a group that is firmly nested within Dinosauria. This is better shown with my personal favorite, the cladocircle, than a cladogram. So as you can see here, this is Dinosauria. If we zoom in on Avelae and look at Avelae in particular, you can see Neornithes is within Avelae, meaning that Neornithes, modern birds, is firmly nested within Avelae, which is firmly nested within Dinosauria. Ta-da! This also helps to explain the so-called problem if humans are evolved from monkeys, why they're still monkeys. It's not that humans evolved from monkeys, it's that humans and monkeys share a recent common ancestor. Some of that ancestor's descendants evolved into humans and some into modern monkeys. Ta-da! Also, humans are monkeys depending on what cladistic group you decide are monkeys. If you decide that monkeys are a clade, simiforms, or a paraphyletic group, all simiforms not in Hominoidea. Anyway, if you consider monkeys to be a clade, then humans are monkeys, ta-da! Humans are definitely apes though, because humans are part of Hominoidea. So stop! Also, cladistics is just a useful tool to test evolutionary history. Hypothesis: are birds dinosaurs or not? Test it using the simplest possible explanation for the distribution of traits within a set of data. You find that birds most simply lie within Dinosauria. You can also use it to test the evolution of characters themselves. Given a well-established evolutionary tree like that of stem birds, how did feathers evolve? Looking at the fossil record evidence for feather-like integument in stem birds, we see that feathers are really common in the clade of Tyrannosaurus plus Neornithes, which is called Coelurosauria. We also find similar structures in pterosaurs and similar ones in ornithoceans. So what's the simplest explanation for this distribution of similar integument in this group? Well, what's the group that includes all of these things with this similar integument? Well, as you can see here, it's Apometatarsalia, the group of pan birds. It is much more likely that this structure evolved once at the base of Apometatarsalia than that it evolved 3 separate times, especially since this structure is hard to fossilize and no genetic evidence contradicts this hypothesis. Ta-da! Similarly, cladistics makes examining convergent evolution easier. Did warm-bloodedness evolve twice or just once in birds and mammals? Twice. The answer is twice. They don't achieve endothermy in the same way. And it makes more sense that it evolved twice than that animals had it originally and then lost it and then got it again. Yay, convergent evolution! But most of all, this is not arbitrary. Is Archaeopteryx a bird or reptile? Who cares? It's part of Avelae, which is a part of Dinosauria, which is a part of Sauropsida. It's definitely a stem bird. It's definitely not a crown bird. Whether or not it's a bird, who cares? It's up to you. Yay! No identity crisis for Archie! Yeah, cladistics is a little bit messy, especially while we work it all out, but it's a model. A model in science is our best guess for how the natural world works. If we got it exactly right, it would cease being a model, so we just try to do our best. Cladistics is our best model for how life has evolved over time. It groups organisms based on how they're related to each other, not based on traits we chose. Some groups get names and others don't, and that is a little arbitrary, but none of the groups are. Frankly, science is messy. If you want a neat and tidy explanation for our universe, you shouldn't go to science. And with every cladistic analysis, every genomic study, we get closer to the answers we seek. So don't give Archaeopteryx an identity crisis. Say no to Linnaeus. Use cladistics. Archaeopteryx and all scientists will thank you. The end.